One of the benefits of this format actually is that more people can participate than usual. So I think in years past, there were maybe a couple hundred attendees. And this year, we're already over 400 people that have registered to attend. So that's really great. And I thank everybody for coming virtually to beautiful Atlanta. It's actually pretty muggy and supposed to rain a lot. So maybe this is a better format this week anyway. Just one more thing I want to say. I want to thank Margie at Rice University, without whom none of this would have happened, even in this virtual format. So thanks to her. And I think now Krastin will say a few words. So Krastin, please.
Yes, I just want to welcome everyone. This is the 10th annual meeting of the iPoLS network. We at NSF are very excited about what you guys have done over the past 10 years, together with our German and French colleagues. We have actually expanded tremendously, starting initially in San Diego in 2011 with about 30, 40 participants. Now, as was mentioned, there are more than 400 people registered. In the next 10 years, in the next decade, of course, we hope to expand around the world. We do have some connections here and there, but we would like to develop a more stronger relations with funding in other countries of the network, and also liberalize the network in the United States and in other countries, so that not only the iPoLS nodes are a part of the network, but also this network serves as a resource for people at all institutions, all graduate students who need support and will participate in the network. So again, congratulations for the great success. I look forward to the next decade, and welcome to the meeting. Thank you very much.
So I guess it's my turn. Hi, my name is Herbie Levine. I'm a faculty member at Northeastern, also a member of the Center for Theoretical Biological Physics, a National Science Foundation funded center working on applying physical science concepts to biology. As Krastin said, this network was formed 10 years ago as a way of trying to create a community of students working on applying physical science to biological problems in many institutions, and not only limited to the United States, but also with a number of international partners. We started small with sort of 4 or 5 institutions that at that point came together to sort of formulate the idea of creating this network, and we've grown in terms of numbers of institutions, in terms of international contacts. And as Krastin says, I think we're looking forward to sort of Network 2.0 for our second decade, where we will have a more broad penetration into many schools. The difference is 10 years ago, there were only perhaps a relatively fewer number of universities that really understood the great possibilities of applying physical science to biological systems. And those were the nodes that started the network. And now I think this idea has been a great success. It's caught on sort of everywhere now. Sort of most major research universities in physics, chemistry, math, computer science departments have people who understand that their techniques and their ideas can bring something unique to the study of biological problems. So with that, I welcome everybody. This is a little bit unusual in the sense of doing this online, and just to mention that we have, in addition to the annual meeting, we have ongoing virtual events, and especially for students, we have funding available if you're interested in finding partners at other institutions and visiting them, hopefully in real instead of virtual space at some point in the future. We actually have funds to sort of foment collaborations between different people working in different universities on related problems in biological science. With that, I'll turn the mic over to Jose, who will also talk a little bit about that.
Okay, I want to welcome everybody in the name of Rice University and myself and Margie that here at Rice, and we are one of the leading nodes of iPoLS. And I want to point out on this, with this short talk, to say that although PoLS has about some institutions that are official members of it. We're open to graduate students in the area of physics of living systems all over the place. So even if you don't belong to one of the nodes, you should become part of our activities. We have our annual conference, we have meetings all the time, so you should contact Margie or myself, and we really welcome all the other people. We want to make the networking compass, all the students on that. Even if you don't belong, you should come and become part. We want to make sure the network becomes diverse. We are on a very challenging time in terms of discussing diversity and broad participation in this country, and the network is really going to focus on that. So to make sure that we have a broad participation in race, gender, minorities coming to it. But more than anything else, I want to say that if you register to this meeting and you are not from one of the nodes, don't be your first time, be part of it. Go to our website at pols.rice.edu. There are several activity seminars going on. Right now, they're all virtual as we have in life, but you're going to have live meetings and stuff like that. They would like to have many, many students that are not belong to the institutions to come. We have support for that. Maybe if you're a graduate student in other areas, you may be interested in the research that's happening in one of the nodes of PoLS. So there's a mechanism bringing you to collaborate with some people in the PoLS of node. So let's make this network broad. If you're not belong to a node, like I said, you're welcome. Join us. If you need more details, contact me or contact Margie. Always go through Margie, it's easier because all the information gets done. But you're welcome, and let's go ahead and have a great meeting. So with that, I think we can start, right?
All right. Yeah. Thanks, guys. We have a great lineup of speakers for this first opening session on biomolecular processes. So it'll run from 10 to 12, and we have seven talks. And a couple things just want to note. So the talks will be 12 minutes with an additional 3 minutes for questions allotted. If you have a question, please use the Q&A box that you should see probably at the bottom of your Zoom panel. And then I will try to sort the questions. You can upvote some if you like, and then I will ask the questions to the speaker in those 3 minutes. That being said, of course, it's not a lot of time to discuss. So there is, I think you should have received in one of your emails, a link to a spatial chat room. And we've asked the speakers to all be present there at the end of the talks at noon. So if you would like to join there and ask them more questions, that will be available. Okay, so with that, our first speakers are Christian Gebhardt and Ganesh Agam from the LMU in Munich. So please, Christian, take it away.
Okay, thank you for the introduction. It's an honor for me and Ganesh to start this nice digital conference. And we want to talk about angstrom precision distance measurements and dynamic detection in proteins with single molecule first resonance energy transfer. And therefore, we actually, built up on a study which was already published 2 years ago. So what we actually want to do in biophysics is we want to detect biomolecular processes by measuring distances and distance changes of fluorophores attached to biomolecules. And this works with this so-called first resonance energy transfer, which is highly distance dependent. And what was shown 2 years ago that in DNA, where we have well-defined structure with well-defined distance, and we can label a donor which transfers the energy to an acceptor, we can have this as a sample system which is robust, static, and we can nicely label specifically the fluorophores. And there, about 20 labs showed that we can reproduce very well with a standard deviation of about plus minus 2 angstrom the distances. And also if we compare to models of the fluorophore distance, we are only a few angstrom away. The worst one was 5 point something, but overall they were even better. And we raised now the question, what if we go to proteins to dynamic biological systems, because there we face way more problems. Like on the one side, proteins are intrinsically dynamic on different timescales. We have to deal with common problems like sample aggregation, sample stability, then also that we label stochastically. So the donor and the might be interchanged, which changes the environment. But then also we have to deal with that the label might introduce malfunctionality, the label efficiency is often not that good, and then also things that the fluorophore properties change because it sticks to the protein, it's quenched or sometimes even enhanced. And therefore we selected the maltose binding protein of E. coli as a model system because it's well studied, it's well known, and we have a rather simple mechanism of these two rigid domains, which are connected by a flexible hinge. And then if it binds maltose, undergoes a conformational change from an open to a closed conformation. And out of that, we selected 3 residue pairs where we have long distances down to short distances, which either are constant like this one here or undergo a change from longer to shorter distance when it binds maltose. And when we do the thread measurements on those mutants, we see that we can then reproduce this conformational change from long distance, short efficiency in this case, to a higher efficiency when it binds maltose, so if the concentration increases. And also for the other 2 mutants, we see here reverse process, or here we have short distance, which just stays constantly high. And having this, we then asked quite a few other groups to measure the systems, which was more challenging due to sample shipping, cooling, sample stability, and so on. But in the end, we managed to get not completely 20, but almost results back. And then what we saw is that we had a good agreement between the different labs for here without maltose and maltose and the 3 different states from left to right. So we could reproduce more or less what the DNA study showed and had a standard deviation of about 2 angstrom, slightly worse than before, but in the same range. And also the accuracy was a few angstrom compared to the model of the dyes and the protein. And one was about 6, the rest was even better. And then as the next point, we asked the groups to check if they can identify dynamics in the proteins, which is the interesting part, since it's not as static as DNA. And therefore we asked them to do on the one side burst variance analysis, where we check the photon statistics, if it's just Poisson distributed, shot noise limited, or if we can identify dynamics in the statistics, which would then deviate from this black static line. And alternatively, checking if we see a deviation, if we compare thread efficiency to the lifetime of the donor, which also for a completely static system should lie on this black line, but in dynamics it deviates. And what we saw is that it's more or less lying on this line. It might slightly deviate, but at least it's more or less in this range. And then if we ask the groups, they also reported, so these are the numbers of the groups. Not everyone was able to do this analysis, but the ones who were able reported with an overall agreement that Mali doesn't seem to be dynamic. Here we have to note that especially this high thread value, some identified as dynamic, which was more challenging because bleaching effects were quite strong. And the second point is what groups identify as dynamic, because in the end, every protein is dynamic to some extent, and therefore it's the question what is considered to be dynamic and what not. And to have a bit of better system on the dynamics, we then also introduce a second system, which Ganesh will show you now. So I will stop and hand over to Ganesh.
I hope now you can see the slides. Yeah. First of all, I would like to thank organizers for giving us wonderful opportunity to discuss our latest and exciting results. And thanks for the Christian to introducing the system and the studies. And as Christian mentioned, that why we chose this particular protein, apart from the first system, where as you can see strikingly, that this protein has two structured domains, but attached with a flexible linker as compared to the first system, where the linker was kind of rigid. And that was the main challenge, that whether we could see this highly dynamic system studying with the other groups as well. And in apostate, this due to this flexible linker, these domains have highly dynamic, and there's a lot of conformational changes going on. When it binds to RNA, the domains are relatively stabilized, and then we see the dynamics decreases. And previously we have actually studied this system where with APO, with the given positions of the labeling dyes, we saw for APO like 75% fret efficiency, but when it binds to RNA and the domains are quite stabilized, but a bit far apart, giving rise to a fret efficiency of 44%. But if you notice, there's also leftover from APO, meaning that this population has not bound to the RNA. And with this interesting system in hand, what we asked whether the groups could detect the dynamics and report, and what is the accuracy and the consistency in reporting the dynamic analysis. But before going into the details of the dynamics, first we compared how exactly the fret histograms looks like when we compare from all 8 groups. So we sent this protein to 8 labs because it's quite challenging to study the dynamics, as well as it's a RNA binding protein, so RNA might degrade and complicate the things. But nevertheless, what we got is, here I'm showing the individual fret histograms, and this is combined analysis, and this is what we measured. So when we did the analysis, what we saw is the fret peaks are fairly similar what we saw, and the deviation is like up to maximum of 5% in all the 3 states what we compared from the all 8 labs. But interestingly, what we observe is that there are few incidences where we saw that there is clear deviation from the rest of the groups. So to find what is the reason for this deviation, for example, here you can see that the fret histogram is quite changed from the rest of the groups, and also here in one of the lab, the leftover of APO fraction has increased. And we asked them to send raw data to us. We reanalyzed ourselves, and what we found is that the deviations are decreased, and the uncertainty in the fret histogram is also decreased as compared to what we measured. As you can see that as compared to what we got from them, and after reanalysis, it is quite comparable what we measured here in with our setups. So indeed, the reanalysis helped, and what we learned from the reanalysis is that the deviations in the fret histograms were majorly due to correction factor deviation that basically controls how the shape of the fret efficiency histograms, and the fraction in one of the lab might be due to measurement conditions like temperature and ligand concentration, because RNA being a ligand, it can degrade, and the APO leftover can increase as if there is no RNA to bind. But the main question what we asked with this protein is how consistent the dynamics across the labs. As Christian has nicely explained how exactly we look for the dynamics in the system, here with second system, what we saw with the BVA analysis, that there is clear deviation from this hemicircle in both APO and holo state, indicating that presence of the dynamics between these 2 states. And also parallelly, fret efficiency versus donor lifetime analysis indicated clearly a shift from this static line for APO as well as holo, indicating that there is indeed a presence of dynamics. And we asked all the 8 labs what they saw with this system with their dynamic analysis. And that was quite encouraging that the 8 labs which studied the second protein correctly interpreted the dynamics, presence of dynamics in this system. But interestingly, they have also correctly indicated that there is no dynamics in the first system. So these results clearly indicates that no matter which setup, in which country they have measured the protein, indeed one can differentiate between presence of dynamics and no presence of dynamics. So that was quite encouraging analysis. And with that, actually, I would like to conclude the talk. So what we learned from this global study is that indeed FRET on proteins can be as accurate, as precise as on DNA with similar uncertainty, even though it has many challenges. And gamma factor, which corrects for the detection for donor and acceptor fluorescence, is indeed a major factor which corrects the shape of the FRET histograms. And the qualitative analysis, whether there is presence of dynamics or not, is definitely a reliable tool to detect the dynamics in the protein with FRET. And as an outlook, what we learned from the study is that if one wants to accurately quantitate the dynamics with FRET, one should at least have 2 diapairs in hand on the with the system. And the choice of the system should be such a way that the system should have a defined dynamics which can be tunable with the given measurement conditions, like for example DNA hairpin, where one can tune the dynamics with the magnesium chloride concentrations. And with that, I would like to acknowledge Thorben Cordes and Christian Gebert for collaborating the study. From our group, Don, Anders, and Simon, and contributors from all over the globe, from United Kingdom, US, Belgium, France, Germany, and so on. And these are the affiliations from all the group participate in the study. And thanks for your attention, and I will be happy to have questions.
All right, thanks a lot, guys. So we ran over a minute or two, but I just want to remind the participants, if you have a question, you can ask it in the Q&A. You can type it in the Q&A box. For any faculty have questions, I think some of them have been granted permission, don't abuse it, to speak. So if anybody wants to speak up, please go ahead with the question. So actually, I have a more general question. I kind of, in looking at all these FRET experiments, how easy is it to figure out where to place the labels? Do you have to do a lot of trial and error, or are you just so good at it that you get it right the first time?
So I think it's not that easy. There are systems where it's rather you can place it almost everywhere and it works quite good. And with others, it's really hard to find a good location. And this is also a major challenge in FRET, designing the right labels where you don't have any interaction on the protein, and also the other way around, where the fluorophore can be considered as free and not influenced by the protein environment. So sometimes it's really, really challenging. And yeah, so this is one of the most important points in our daily work, I would say.
Okay.
I have a very short question. It was a very, very interesting talk. I just like to know 2 very short things. One is, how about the dynamics of the protein? The protein is moving a lot. If you don't have a status of fixed distance, what exactly you're measuring? How do you put this time scale to the process? And second, I agree with you, you do better with 2 probes, but you do even better with 3 probes. So can you exactly quantify a little bit more to say what you gain by going from 1 to 2, and what happens if you put more probes?
So Christian, you want to go ahead, or I can?
Yeah, I mean, you can take the first one maybe.
Yeah, so regarding the first question of about dynamics, to compare the dynamics on a second protein, which was why we chose this protein. It was quite interesting to see underlying dynamics, because for first protein, it is the dynamics are on the second time scales, but in second protein, dynamics are on the millisecond time scales, like sub-millisecond time scales. So definitely there are analytical tools to detect different scales of dynamics. For example, as we shown, one of the method that we use, BVA, which basically checks the variance in the fret efficiency within each burst. There you can have the feeling of the dynamics if there is change in the fret efficiency within few milliseconds within the burst. But to have the define dynamics in a better way, we rely most on donor lifetime as well, where we check the donor lifetime on nanosecond time scales, where one can define the states of the dynamics depending on the donor lifetimes, which are on the nanosecond time scales. And yeah, there are other tools to analytical tools like species FCS, where one can select some of the species from the fret efficiency fret histograms and look for the correlations more carefully and so on. So yeah, it's a bit tricky. That's why if you want to compare the dynamics, one should have a system, for example, DNA hairpin, as I mentioned, where one can tune the dynamics with simply by the measurement conditions. Like, for example, for DNA hairpin, it's like magnesium chloride. You can tune the dynamics, and that could be a better to compare better, because what we found is also if you change the dye, some of the dynamics also changed a bit. But yes, of course, when you compare 2-3 dye pairs, then one can delineate at the actual dynamics with the given methods.
Thank you.
Yeah.
All right. Thanks again for the great talk. We do need to move on, unfortunately. So any questions that were not answered, just a reminder, you can ask those in the spatial chat afterward, and the speakers have agreed to be there at noon Eastern. So for our next talk, we have David Garcia from Maryland. So David, please take it away.
Yeah, thank you so much for the introduction. I would like to first thank the organizer for giving me the opportunity to talk here at IPOLS. I'm a physics PhD student at the University of Maryland in the PoLS lab and a predoctoral fellow on the National Cancer Institute in Gordon's Hager lab. And today I would like to talk about transcription factor dynamics, their interactions with chromatin, and how these affect gene expression. First, we need to talk about the central dogma of biology. So genes, all the information in the cells is stored in DNA. This DNA is transcribed into RNA. The RNA is then exported to the cytoplasm, where it's translated into protein. And then a gene's level of expression reflects the amount of its RNA in the cell. And the interesting part about this is almost all cells in our bodies, they are genetically identical. And the difference between them is just they have a different gene expression profile. An important player in the transcriptional regulation are transcription factors. These are specialized proteins that they bind to specific sequence of DNA and they regulate the expression of the genes. And depending how they interact with chromatin, they can be classified in different families. First, we have complexes, so transcription factors that they interact together and they bind to different places in chromatin. Then we have pioneering factors that they open chromatin by an ATP-independent manner in a process called wedging. So basically, they bind to close chromatin and they remodel chromatin. Then we have dynamic-assisted loading, where transcription factors bind to close chromatin, then they recruit remodelers, which they'll modify chromatin in an ATP-independent manner. And finally, we have transcription factors that they play an architectural role. They usually have a really high binding affinity, and they are responsible for stabilizing loops and give the topology to chromatin. So in our labs, we are really interested in understanding how these transcription factors interact with DNA, organizing chromatin, how they intranuclear dynamics of these transcription factors, how they bind and bind from DNA, affect gene expression. The problem in the field is that most of the studies that are trying to understand transcription factors, regulation of gene expression, they are being a static analysis. So a popular assay is something called ChIP-seq, chromatin immunoprecipitation, where you fix the cells, then you immunoprecipitate the transcription factor of interest, and then you sequence the pieces of DNA where the transcription factor was bound. And then what you're gonna find is the intensity, high intensity of the sequence where the transcription factor binds. And then if you want to understand the gene expression profile of a particular population, you kill the cells, you extract RNA, and then you quantify the levels of RNA. But since we're interested in the dynamics of the transcription factors, we're gonna use one model that we like a lot, that is the glucocorticoid receptor. The glucocorticoid receptor is a hormone-inducible transcription factor, which is really important for development, metabolism, immune response, and cancer. The interesting part about this transcription factor is that it is usually in the cytoplasm, and when you add the hormone, it translocates into the nucleus where it will regulate gene expression. So we're gonna use the glucocorticoid receptor to understand the dynamics of transcription factors and how these dynamics affect gene expression. And to do this, we're gonna use single molecule tracking, where we take the transcription factor of interest and then we add a protein, a halo tag protein that has a similar size of GFP. And then it has a ligand where we can then incubate it with an organic fluorophore, and the organic fluorophore will covalently bind to that ligand. So we introduce the tag protein either by a transient transfection or a stable, or we just make stable set lines. The day of the experiments, we incubate with the fluorophore of interest. Then we extensively wash to remove any unbound fluorophore. And then we do high-low microscopy, which is similar to TURF. You just increase the angle slightly. And this helps to remove background fluorescence. So this is a sample of the glucocorticoid receptor after the addition of dexamethasone, which is a synthetic ligand, pretty common in the clinical field. And as you can see, there's different types of kinetics, transcription factors moving, diffusing around. Some other ones are binding to chromatin. And then if we fix the cells, of course, all these kinetics are lost. And this is where most of the genomic experiments are done. And depending on the protein that we analyze, are going to have different types of kinetics. For example, here I'm showing halo H2B, where they are basically just incorporating into chromatin, and you can see just the jiggling of chromatin. Then we track the transcription factors, and then we classify them either in bound or unbound. If we look just into the bound molecules, then we're gonna, if we make a chymograph, they're gonna look like stripes over time. So a transcription factor binds to in at time T1 and then unbinds or photobleach at a time T2. Then we are gonna assume that the system is ergodic, so we can align all the trajectories, and we're gonna have then at the distribution of the time that a transcription factor stays bound to chromatin, and we call this the dual time distribution. This is a sample of the dual time distribution for the glucocorticoid receptor, and then we feed it to a double exponential feed, which is the most popular model in single molecule experiments of transcription factors, but this doesn't fit to our data. The double exponential feed is really popular, where the slow component represents a specific chromatin binding, and the fast component represents non-specific chromatin binding. What we actually saw is that the dual time distribution is power law distributed. We did experiments as well with the synthetic hormone, that is dexamethasone, is more powerful. And what we saw is actually the transcription factor binds for shorter times compared to the natural ligand, and it was really surprising. We can deactivate the transcription factor by washing out the hormone, so the transcription factor won't bind to chromatin anymore. And what we saw is that it still binds, but with faster binding times, and it still follows a power law distribution. We looked as well at different families of transcription factor. In particular, we look at pioneer factor FOXA1, which is supposed to interact with the estrogen receptor based on genomic experiments, and they follow basically the same type of kinetics as we would have expected. Something similar happens with the remodeler SMARCA4, which is supposed to interact with GR. And again, they follow the same types of kinetics as we expect. If we look at structural transcription factors like CTCF, which they are supposed to have a really high binding affinity, they still follow a power law distribution, but with longer dwell times. So we came up with a model to try to explain this power law distribution. So these transcription factors bind to thousands of places into chromatin in different microenvironments. So each microenvironment and each binding site is going to have a distribution of binding affinity, and then we're going to have a broad distribution of binding affinities into the nucleus. We can think this as the energy landscape to be rough on chromatin. And then we can model this, and then we can find that if you have this broad heavy tail distribution of binding affinities, we're going to have a power law distributed dwell time. But then we want to correlate these kinetics to gene expression. And to do this, then we took the MMTV gene, which is a gene that is regulated by the glucocorticoid receptor, and then we added stem loops. These stem loops, when they are transcribed, the fluorescent protein is going to code them, and then we can see transcription happening in real time. So this is an example. And what we're going to see is basically bursts of RNA production. And this burst of RNA production is where the high intensity happens. We are going to call them the on times. Then we're going to have periods where the transcription, where transcription is not happening when the gene is off, and this is going to be off times. Then when we calculate the on times, we saw that the synthetic hormone has shorter dwell times compared to the natural hormone corticosterone, which correlates pretty well with the dwell time distribution that I showed previously. Another good model to try to understand how the kinetics of transcription factors affect gene expression is some mutants that we have in our lab, the GRDIM, that is a dimeric form of the glucocorticoid receptor, and GRMON, that is a monomeric version of the glucocorticoid receptor. The wild-type version, when it binds to DNA, is usually tetrameric. And what we found is if we compare GR wild-type and GRDIM basically binds similar to the GR wild-type. This is ChIP-seq, and we're quantifying the amount of binding of the transcription factor. And the GRMON, basically, we don't see any binding. Then we did RNA-seq, and what we saw is that GRMON basically is a dead receptor, as expected, since it's not binding to chromatin. But GRDIM was really surprising because it was binding to chromatin, but the response was really low. So we had a dramatic reduction in the gene expression for GRDIM. Then we went to do SMT experiments on these mutants, and what we found is that the GRDIM and the GRMON, they bind for shorter times to chromatin. And this explains then why we see such attenuation in the gene expression of GR, since the transcription factor cannot bind long enough to recruit all the machinery for transcription to happen. Then the conclusions is that different families of transcription factors present power law distributed dwell times. A broad distribution of binding affinities might be the responsible of heavy tail distributed dwell times. A specific and unspecific chromatin binding is power law distributed. GR dwell times are correlated with on times of RNA bursting. Chromatin binding by genomic studies is not a sole predictor of gene regulation, in contrast of what the field used to believe. And the missing link between gene regulation and chromatin binding might be the time that a transcription factor is bound to chromatin. Then with this, I would like to thank my advisors, Professor Arpiro Paya at the University of Maryland and Dr. Gordon Hager at NIH, everyone in the team that helped with all the experiments and are involved in the project, all the members of the two groups for all the helpful discussions, and then our collaborator, Christopher Jasinski, the imaging facility for helping us with all the SMT experiments, and my funding agency. And I'm happy to receive questions.
All right. Thanks a lot, David. So that was, not only was that a wonderful scientific talk, that was a master class in nailing the timing perfectly at
Oh,
12 minutes. So we have a question in the Q&A from Songjim Kim, who asks, is transcription factor binding and rebinding kinetics affected by nucleosome density?
We believe so. We believe that the microenvironment affects the binding kinetics, and in particular, the density of sites and the density of the transcription factor. The problem is, as we are doing single molecule experiments, as I show, we're just looking at the whole nucleus, so we cannot distinguish these microenvironments. However, there's other techniques, and you can go in this paper where we quantify the binding to a specific site. But to be able to really understand if it is affected by the microenvironment, they will have to do transcription factor kinetics and a specific gene.
Hi, could I ask you a question?
Yeah,
this is Dave Tarumulai.
Okay.
It was a fantastic talk. It seemed like you were using the trap model that people in the glass field have talked about for a long time to rationalize the polar distribution. I was wondering if you have any structural feel for why you have this dwell time distribution in some wells and the dwell time distribution in different wells, in fact, are distributed in such a way that you actually get this power law.
That's a really good question. So basically, when a transcription binds to chromatin, it will bind to a sequence of DNA. And the interesting thing about these transcription factors is that there's just no one particular sequence of DNA that they bind. So we think that the binding affinity of these transcription factors is gonna be related to the sequence that we call the motif. So here what we are showing is basically each motif is gonna have a different binding energy, basically. So actually in a paper that we have in preparation, we quantify the distribution of the motif strengths for these transcription factors. And what we saw is that if the transcription factor has a broad distribution of motif strengths, it follows a power law distribution. If you do mutations on the proteins to narrow the distributions of motifs strengths, then you recover a double exponential distribution.
Thanks.
Hello. This is Peter Wellness. Very, very nice. Actually, I think this is a very, very deep problem, and it's one that we've come across in the problem of NF-kappa B and some papers with Vít Potoyan about it. But that's it's very exciting, your results. You know, this debate between double exponentials and power laws has been going on in general in biological physics for quite a long time. And even in the ordinary dynamics of biomolecules when Hans Frauenfelder first found it. Now, he had a real great experiment which showed that you had a heterogeneous distribution, and at least people who understood it were convinced. And that was what was called the double jump experiment, which is that you let something dwell for a while or something like that, and then you repeat the signal. So for example, if it's the addition of a hormone, you would add the hormone, let the material decay for a while, then add the hormone back so it binds again, and then see whether the guys that have already fallen off again fall off rapidly, or whether they go back to the random distribution. Have you tried that experiment?
We tried a similar experiment where we wash out the hormones at different time intervals. And we keep seeing power law distributed dwell times.
Right, but I think the double pulse is really what really clinches it. So if you read back in the old 1970s papers by Frauenfelder, you'll see something called, I think it's called the double pulse.
The double jump. Yeah, another problem with our model. The problem with our model, and I think, that's an experiment that we should pursue. The problem is with the glucocorticoid receptor, we cannot do that. Because when we activate the receptor, it goes to the nucleus, we watch the hormone, and when we try to reactivate it, we cannot reactivate the receptor.
Oh.
And we still don't understand the biology behind that. Why after we remove the hormone and we try to reactivate the receptor, it basically just doesn't work anymore. So we'll have to move to another transcription factor that we can do pulsing of hormone of activation. But
Okay,
thanks for the
very
suggestion. Yeah.
All right, so now we are running a minute or two over, so I think we need to move on. So thanks again, David, for that. And I think you'll have a few more questions in the spatial chat afterwards. So for our next talk, we have from Georgia Tech, Michael Ryan. So Michael, please go ahead.
Can you see my slide, JC?
Yes, I can see it. I think everybody.
Wonderful. Let me reset my timer so I'm not going to go over. So hi, my name is Michael Ryan. I work at the Georgia Institute of Technology in the Harold Kim Lab. And our lab specializes in single molecule DNA studies. Today, specifically, I want to talk to you about the flexibility of DNA nucleotide mismatches. Now, just to make sure everyone's on the same page, we'll start with a refresher. DNA, it's a double helix, made of two individual strands that are held together through hydrogen bonding. On the right, you can see that nucleotides have preferential partners. G and C pairs share 3 hydrogen bonds, while A and T pairs only have 2 hydrogen bonds. Additionally, the bonds tend to line up very well between the 2 nucleotides. So this raises an interesting question. What happens if the nucleotides are mispaired? And sure enough, when you pair nucleotides that don't match, they end up adopting some odd interactions. They have to twist, shift, slide, and reorient to find a new minimum energy state. I put my favorite mismatch in the center here, by the way, go Georgia Tech. And what these alternative conformations do is they disrupt base pairing between the strands, and they also disrupt base stacking between neighboring nucleotides on the same strand. So mismatched DNA alters both the sequence information as well as the structural integrity of the DNA strand itself. So understandably, cells want to correct for these kind of problems. And there's plenty of studies recently exploring the mismatch repair system present in a host of cellular life. Interesting thing that's been noted by many groups is that when these mismatch repair proteins couple with DNA, they're found to bend the DNA very sharply. So it's been suggested that maybe a weakened helical structure caused by a mismatched nucleotide may play a role in both the recognition and the repair of those mismatched sequences. So for reasons of both fundamental research as well as application, groups have sought to characterize the flexibility of DNA for sharp bending angles and for mismatched sequences. And they mostly come in 2 flavors. You'll have molecular dynamic simulations, and you'll have crystal diffraction studies, where the simulations explore free DNA while the experiment tends to look a little bit more on protein-coupled DNA. So problematically, though, these 2 styles don't completely agree with one another on the various parameters that they find for the DNA and its mismatches. A more recent paper published this year sort of succinctly describes that, saying it's not clear whether a single set of parameters can represent both unconstrained, it's free DNA, and protein-coupled DNA at the same time. In this publication, they had to develop a hybrid model which merged parameters from both fields of study. So there's a divide between simulation and experiment, as well as between free DNA and bound DNA. So I'd like to try and bridge the gap a little bit by experimentally measuring free DNA. So DNA strands explore a wide conformation space, and this is fueled by thermal energy kicking it around. We can use this to our advantage because bending DNA strands is not a very easy task. When the DNA strand explores a sharp conformation that's sharply bent, we want to try and capture it in that state so that we can measure its bending dynamics. And additionally, we need some way to visually detect these conformational changes because DNA is terribly small. So let me propose to you the solution to problem 1. This is a DNA strand designed such that it has complementary ends on the left and right. When the DNA strand bends enough, explores a conformation that brings these ends together, they can hybridize, forming a double helix down on the bottom here, and that will temporarily lock the loop into place long enough for us to make a measurement. And additionally, each strand is designed such that we can insert a base pair on the top and the bottom to engineer whatever mismatches we want to test. Now, as for detecting these changes, I'm going to mention to you, which should be a familiar tool now. Christian and Ganesh mentioned it in the first talk, fluorescence resonance energy transfer, FRET for short. And essentially the way this works is that you'll shine a laser across two dyes. Only a green donor dye, in our case green, donor dye can absorb the energy and begin fluorescing. But if it's requisite red donor dye, or excuse me, if it's requisite red acceptor dye is brought close, the donor will transfer its energy to the acceptor. The green fluorescence will disappear and we'll start seeing red. So it's like a switching interaction based on the distance of the dyes. So you can take these two dyes, put them on either ends of my DNA molecule, and when it's in an unlooped conformation, you will see green fluorescence through your microscope. And when it's in a looped conformation, the dyes are brought close enough together that we get that FRET interaction, and you'll begin to see red. So here is a video of that happening. This is the switching interaction. The field of view here, both of them observe the same location in space. They're just separated by wavelength. So green on one side, red on the other, but coming from the same point. You can separate it with some dichroics and filters. And what we're watching here is a single DNA molecule looping and unlooping in real time. So you can do this. You can do it for a wide field of view for your camera and pick out where all the points are and start to build up some robust statistics. So you take all these blinking lights and you put them into a fluorescent time trace like this. You can see the switching back and forth. And you sort it all down to a binary state trace, where the top one would represent looped, the bottom state here represents unlooped. And you can extract out the dwell times of this, how long your molecules spend in each state. When you do this for hundreds of molecules, you can throw it into a nice survival curve, fit an exponential decay, and you can pull out some rate constants from that to understand how the dynamics of this are happening. Now, it's important, though, to talk about expectations before I show you my data. We had to think about what does the theory and models say that we should see from these results. So it's beneficial at this time for me to point out to you that DNA is a semi-flexible polymer. Now what this means is that there is a stiffness associated with it called the persistence length. If a polymer is longer than its persistence length, like on the right here, it is likely to be contorted, looping, and twisted. It's very much in an entropic regime. A polymer, however, that is shorter than the persistence length is more like an elastic rod. It's definitely an energy-dominated regime. Our molecule in this experiment is 105 base pairs, and the persistence length is 150 base pairs. So we're well within this energy-dominated regime where we would expect our DNA strand to act more like a rigid spring. And this slide does a good job hammering home that intuition. If a rigid spring in this case is difficult to bend, requiring a lot of energy put in to bring the ends together, it should be easy to unbend as that high restoring force works hard to straighten the molecule out. So intuitively, we would expect an anti-correlation between our looping and our unlooping rates. But we can also explore this mathematically as well. Unfortunately, with the time constraints, I'm not gonna really be able to go into how you can derive these relationships, but the intuition follows from the last slide. Our looping rate is dependent upon how much bending energy you have to put into the molecule to bring it to this transition state where the ends are close together. And the unlooping rate is dependent upon the force that is trying to unloop your molecule at that transition point. So all we have to do then is have a bending energy model, and we can start to predict what the rates would look like. And a very popular one is called the worm-like chain model. And it defines a long chain full of a bunch of elements that have a bending stiffness associated between each element. And in this case, it's a harmonic bending potential. This is a very popular model used in DNA studies. So we can take this and we can toss this bending model into an energy minimization program. So what it does is it takes these chains here. You can see some representations, and it makes small adjustments to their angles and positions until it finds a minimum energy state for what the molecule would look like. So what we can do then is change the center base pair stiffness and look at how our rates would be affected. What we see changing the stiffness is that it has a positive monotonic relationship for both bending energy and for force. This makes sense. Stiffer bases would create more, you need more energy to bend, and it would also have a larger restoring force. And when you put that into our rate calculations for looping rate and for unlooping rate, we again see that anti-correlation that we would expect. So not only intuitively do we see this, but also from a mathematical standpoint. So now that we've done that, let me point you to my data. Here's a plot of our looping rate, which is a function of the nucleotides that are inserted in the middle along this x-axis. You can see the matched DNA shows the lowest looping rates, suggesting that it's the most rigid, strongest helical structure. And every single mismatch is consistently weaker in structure and more likely to loop and form sharp bends. So based on the worm-like chain model we just discussed, without changing the order on this x-axis, we would expect to see our unlooping rates have a decaying curve. But we do not see that at all. The order of the bases has not changed. There just simply isn't that anti-correlation that we would have expected. And this is true across a whole wide range of salt conditions that we measured. Here's 3 plots for you at 0.3 molar, 0.5 molar, and 1 molar sodium chloride concentration. You can see the Spearman correlation are all very low and represent both positive and negative values. So this data does not show what a harmonic bending model would predict. What our data does tell us, though, is pretty interesting. We tried varying the temperature in our study as well, and we find that the looping rate anti-correlates very strongly with the melting free energy of this mismatch and its 2 neighbors. So what this is saying is that weak bonds make for easier looping. And what this means is that looping appears to have a very strong connection to the mismatch, breaking its bonds and maybe forming bubbles or kinks, anything else that allows for structural change to get sharp bending. And this is not accounted for in the harmonic bending model. What about the topic of DNA mismatch repair? Is there a link to intrinsic bendability? Well, the x-axis here is ordered in decreasing rate according to my data. See the base pairs. And you'll see if there's any correlation between my data and these top rows and any of these other publications looking at binding and repair of the mismatch repair protein, it would go from dark blue to a bright white. Visually, it's pretty apparent that that doesn't seem to be a connection between propensity to bend and likelihood of a mismatch being found or repaired. And I've added the Spearman correlation values along the right side, so you can look at those as well. I'm running low on time. I need to progress. So allow me to synthesize this data down into some usable numbers for you. It's possible to analytically calculate the probability that the ends of your DNA molecule will come together. So we calculated those probabilities. It's called the J factor. And we did that based on changing the stiffness of the middle base pair. Comparing my data to this, you can extract out what bending parameters would be needed to reproduce something like this. And that's what this is here, decreasing relative stiffness in units. These are the values that you would take and toss into your bending model if you were simulating this. But again, like I said, the harmonic model isn't necessarily the best thing to look at. So we propose an anharmonic bending model. And what this allows for is for bend weakening. This is phenomenologically describing the melting or kinking or structural changes that you would see at these sharp bending angles. If you take this anharmonic model and take a look at it in our energy minimization that I was telling you about earlier, we try and find the most optimal shape. You no longer see this anti-correlated decay. You'll now get a range of looping and unlooping rates where you could imagine any point in this space would correlate to different bending parameters for the bend weakening. So 2 more slides, I'm almost done. Just to hammer home the point, there are plenty of simulation groups that witness this kind of bend weakening. Here's a recent publication where they bend DNA molecules with a varying number of mismatches. And they too see a transition at sharp bending, which fits very well with the anharmonic model that I'm suggesting. So allow me to conclude. We designed a DNA molecule and constructed it with a single molecule fret to measure it in real time. These results were counterintuitive and don't follow a harmonic bending model, something like the worm-like chain model would predict. Bending at sharp angles has some relationship to structural change and bend weakening. DNA mismatch repair does not appear to be connected to intrinsic bending properties of a mismatch. And finally, we really ought to look at using some new and expanded models. Harmonic bending works well for small bend angles and for long DNA, but it kind of breaks down at the extremes of short DNA and sharp bending. So I'd like to thank my advisor, Harold Kim, for providing direction and guidance in this work, my lab mates for advice and some valuable discussions, as well as our sponsors, the National Institute of Health and the National Science Foundation. So I'll be happy to take any questions.
All right, thanks, Michael. I think we really only have time for one or two quick questions. So we have a couple in the Q&A. So Joe Beckwith asks, are the N-harmonicity factors purely phenomenological, or can you get them from some sort of first principles approach?
I am not sure how to answer that. I'm sorry.
Well, are you just fitting them?
Well, I haven't fitted anything. I don't have I haven't extracted parameters from my data for the anharmonic model. It's merely a proposition to account for this kind of structural change that we see. I don't have a model to mathematically do that just yet.
Okay, and then we have one more quick question from Ting Wei. Did you do the bending, unbending kinetics by surface anchored DNA? Where is your anchor located?
Yes, so here's a model of what it looks like actually. It has some biotin and it's a neutravidin and a biotin peg on the surface. The anchor point here is, this is pretty well representative of what it would look like on the molecular setting. Yes, so it is on the surface, it's held in one place, and that would constrain the amount of conformations that it could take. But I still think this data is robust enough to draw a good answer from.
All right, thanks for that again, Michael. And sorry to any faculty that wanted to ask questions, but we do need to move on.
I'll be in chat and doing the Q&A if anyone wants to ask any more.
Great. Okay, so our next talk is from Alessandro Cernjar. I hope I said that correctly, from King's
Hello, everyone. Here I am. That would be Cherniar.
I really, okay. Well,
that's all right, don't worry, that wouldn't be the first time. So let me share the screen with you all. I think that that Michael needs to unshare his screen first. Okay, let me try now. Okay.
All right, great, we see it.
Just 1 second. All right. I hope that everybody can see and hear just fine.
Yes.
And in this case, everybody just fine?
I think so.
Okay, fantastic. So I'll start. So I would like to thank you all for allowing me to speak here today. I am Alessandro Cherniar. I'm doing my PhD at King's College London in the UK. And today I'd like to talk to you about key features of the serotonin-gated ion channel. So let me start my talk talking to you about pentameric ligand-gated ion channels. They are proteins that are embedded in the lipid membranes of nerve cells, and they're involved in fast synaptic transmission. Their aim is the regulation of membrane potential through the passage of ions such as chlorine or sodium through structures like selectivity filters and a hydrophobic gate. So their opening, also referred to as gating, is ultimately triggered by the binding of smaller molecules called ligands or agonists. And this mechanism is not fully understood at the molecular level. The serotonin receptor or 5-HT3 receptor is one such pentameric ligand-gated ion channel, and its importance is proved by its involvement in diseases such like Alzheimer's, schizophrenia, drug abuse, and among other things is also the target of anti-vomit drugs for patients undergoing chemotherapy. So this structure is divided into 5 subunits, hence pentameric ion channel, and also into 3 domains. An extracellular domain that is mainly made up of beta helices, as you can see here, the transmembrane domain that's made up of alpha helices, and an intracellular domain that here has been cut off. So the projects I would like to talk to you about today are both focused on the transmembrane domain that is depicted here. And it's made of 4 alpha helices per subunit called M1, M2, M3, and M4. So here we have the M2s depicted in blue, which lines the ionic channel and are buried within the protein. We have then the M1 in yellow and the M3s in red that are partially exposed to the lipidic membrane just outside the protein. And then in purple, we have the M4s, which are the outermost helices and therefore the most exposed to the lipid membrane. Just on top of this transmembrane domain, there's the extracellular domain at the interface between the two of them. There's this cis loop that gives the name to the receptor superfamily, the cis loop family, which is also depicted here in yellow in the previous slide. So what are the goals of my talk today? I would like to talk to you about two projects focused at two interfaces of the 5-HT3 receptor. One such interface is the extracellular domain, transmembrane domain interface, where we found a molecular switch, and the protein-lipid interface. Both of these projects are investigated with in silico techniques, namely classical all-atom molecular dynamics, and one enhanced sampling technique, namely well-tempered metadynamics. Both of them are performed with within the Amber force field family and within the NumD software family. So let me start talking about the first project that is also being reported in the Journal of Physical Chemistry Letters. And like I said, it's focused at the interface between extracellular domain and transmembrane domain. Namely, it's focused in the loop that connects the top of the M2 with the top of the M3. Here we found this proline 281. And the molecular switch I would like to talk to you about is the proline trans-cis isomerization. And this phenomenon consists in the interchange of this dihedral angle that is depicted here in green from 180 degrees trans to 0 degrees cis. Now, why are we interested in this phenomenon? It's because back in the days, in 2005, mutagenesis electrophysiology experiments proved that the channel responsiveness would correlate with the isomerization propensity of proline analogs or other mutants that are substituted at this very position. So what is our goal in this context is to provide an in silico counterproof that this molecular switch is actually part of the overall gating mechanism. How do we do this? We did well-tempered metadynamics simulation for the reconstruction of the free energy surface associated with this process. You can see these two graphs here that express the free energy surface for a control proline dipeptide in water here on the left, and for our proline 281 here on the right. In both these graphs, the isomerization process is described by the x-axis. You can see here that we have in the center the cis basins separated by the trans basins that are on both edges of these graphs. So what do we notice? That essentially these basins are separated by high energetic barriers that in the dipeptide would go up to 20 kcal per mole in both directions of isomerization. And what do we notice in the protein environment? That this symmetry is essentially lost because the right-hand barriers would go up to 25 kcal per mole. So this essentially disrupts the barrier symmetry and potentially creates a preferential direction of isomerization in the anti-clockwise direction. Also, very importantly, this different energetic landscape may be a first hint of a different isomerization rate in the receptor with respect to water. And what does this mean on the mechanistic, on a mechanistical perspective? So flanking proline 281, we have this proline 280, and upon proline 281 isomerization, this residue is essentially able to make hydrogen bonds with different parts of the receptor. When the isomers are in cis or in the transition states, the threonine is able to make hydrogen bonds with this tyrosine 140 that belongs to the cis loop, hence to the extracellular domain. Conversely, when we are in the trans isomer, it would make an hydrogen bond with the leucine 282 that still belongs to the M2, M3 loop. So overall, what does this mean? That basically the proline 282 isomerization is a possible mediator of mechanical signal from the extracellular domain, where we found the ligand binding pocket, to the transmembrane domain, where the hydrophobic gate of this receptor is found. There's also another side note about this project is that we did also molecular dynamic simulation of 4 different models with progressively more subunits with cis prolines. And these models are referred to as 5-trans, 3-cis, 5-cis, and 5-trans, 5-cis, depending on the number of prolines that we have in the cis isomer. So what do we notice is that upon isomerization, the channel that was originally in a closed conformation still remains closed. But there's an interesting structural difference just below the extracellular domain separation line from the transmembrane domain, which possibly hints that the selectivity filter that you would find here would get in a different structural conformation. Now I would like to focus my attention to the other interface I mentioned, and this is the interface between the protein and the lipid membrane. So there's evidence in the literature that membrane lipids can modulate membrane proteins' function and or alter their features. And this does not just happen in pentameric ligand-gated ion channels, but it's true for many other membrane proteins as well. So in this context, we were interested in doing molecular dynamics simulations of 3 models with different lipid concentrations of the membrane. And for this project, we focused on 3 lipidic species, namely cholesterol, that is very important for the brain composition, for the brain cell membrane composition, and also on 2 typical phospholipids, namely POPC and POP. What did we observe in this context is that lipids would form interactions with both with the M2 and M3 loop, namely with 2 residues in particular, with leucine 273, and interestingly, again with threonine 280, and also with the cis loop, that again is part of the interface between the 2 domains. And most interestingly, the lipids are also able to form stable bindings to the transmembrane domain of this protein. Some of these events would lead to partial intercalation of these lipids within the transmembrane domain, sometimes also reaching out for interior parts of the protein, like the M2 themselves, which are supposed to be buried within the protein. And very interestingly, these lipids that we have investigated are able to make these interactions and binding events in a very different way. Like while phospholipids are able to reach for these structures thanks to their length or their flexibility, cholesterol is more able to intercalate within a single subunit, and there's also evidence in the literature that over very long times it can even fully intercalate within one subunit. And lastly, we noticed that in the model that we have with the highest concentration of cholesterol, the number of these stable binding events is enhanced. So this is pretty much what I have to say about my 2 projects, and both of them are hinting that these 2 structures, these 2 loops are very important for the protein function, so the M2 and the M3 loop and the cis loop. And how is this done? Because both the proline switch that I talked about, but also the lipids of the membrane are able to give rise to useful interactions that could eventually pin the cis loop into making into a convenient position for the transmission of the mechanistic signal. So just a quick overview of the projects that I'm taking care of now. I am investigating the effect of an alanine mutation on the cis loop interactions, and we're also interested in investigating serotonin in the binding pocket. I would like to thank my supervisor, Carla Molteni, my collaborators, Sarah Lammis and Suzanne Mezoy at the University of Cambridge. I would also like to thank King's College London, the Thomas Young Center, and the High Performance Computing Resources, Archer and Thomas. And thank to all of you for your attention. I am eager to take your questions.
All right, thanks, Alessandro. So we do have time for some questions. I don't see any in the Q&A, but if any faculty would like to speak up, feel free to.
Oops, sorry.
So maybe I can ask about, I don't know if I caught it, what are the timescale of your simulations typically?
That's a good question. I didn't actually mention it. So for the well-tempered metadynamics, we perform them for 800 nanoseconds for the protein environment, because given the complexity of the environment itself, it's required a lot of time for the convergence of the free energy landscape, while for the dipeptide, 40 nanoseconds were enough because the environment is less complex. In the case of the molecular dynamic simulations, all of them were up to 500 nanoseconds, so alpha microsecond essentially.
Okay. I just have, here is José Almuchig, just a fast question. This transition that you have, is there's a real competition between the energetics and some entropic configuration entropy change. So I'm wondering if by using metadynamics, you may sort of distort the participation of each of these effects.
Sorry, can you ask the question again? Sorry.
Basically, the system here, you have some entropic fluctuations and some energy barriers you have to cross as you do your dynamics. When you do metadynamics, you're lowering these energy barriers. So is this competition between entropy and energy done correctly when you do metadynamics, or you may run into trouble into that?
So basically, you're asking me if the interactions are making
the balance, the competition between the two effects, right? How much just entropic fluctuations versus you're energetically jumping, right? During this transition.
I would guess so. Yes. I'm just trying to open my my presentation again. I don't know what happened, so I can possibly. Okay. So here. So you're asking me if there's a difference between the electrostatic contribution, not electrostatic, the interaction contribution and the entropic contribution for the overcoming of these barriers.
That's right. How much just a fluctuation you have to wait for the system fluctuate to go through, and how much you just jump over this barrier, right? These are two possibilities you can have here, right?
Well, yes, essentially, although the interactions, I would guess that they are much more important in the receptor environment because of the complexity of the environment, like I was saying. There's not just water, like in the dipeptide, but there are also other residues that can or cannot form in interactions, which can help or help not the transitions.
Okay.
Okay, so we have one more question from the chat or the Q&A. So Robert Rosenstein asks, are there situations where quantum mechanics must be used on some portion of the overall system?
I would say so. Although for the most part, I would say that this regards the properties of the ligand inside the binding pocket, because sometimes it's important to capture certain effects of certain interactions for the stability of the system. In this specific case, it depends on what you're interested, because if you're interested in saying if this receptor isomerization can be part of the overall mechanism, you may want to see how it is disrupted with respect to a control system. And in this case, I think that an unsampling technique may be more useful rather than just making quantum mechanical calculations.
Okay, great. Thanks for that and for the nice talk, Alessandro. So our next speaker is James Lyman from Rice University. Yeah, James, and we see your screen, so
yes,
everything is good.
Give me a second so I can make this smaller. There you go. Good morning, everybody. My name is James. I'm a graduate student at the Center for Theoretical Biological Physics. And today I'm going to talk about part of my current research project, which is to understand the role of R2-3 complex, an actin filament brancher, in the dynamics and structures of Actin-myosin networks. And this work is collaborations with my good friends, Carlos Bueno and Yossi Elias, and my PI, Dr. Margaret Chung, along with Dr. Herbert Levine, Dr. Neil Waxham from UT Health, and Peter Walnes. So what are Actin-myosin networks and why do we care about them? actomyosin networks are systems of actin filaments that are mainly reorganized by active myosins or motor proteins and passive cross-linkers. Now, actomyosin are ubiquitous in biology. As you can see here, actomyosin networks control cell shape both globally and also locally. And you see fluorescent images here, examples of actomyosin networks that you can find in muscles and to platelet and fibroblasts. You see that there are multiple actin binding proteins in this network. For example, alpha-actinin, and I am going to discuss more in much details different kind of actin binding proteins that we are interested in. So let me start with a specific example. We are interested in actomyosin networks in the neurons. I'll keep this running for a couple more times, and I'll describe in a second. Actomyosin networks, like I said, are found in the neurons. Experimentally, it has been shown that the growing and shrinking, or the plasticity of dendritic spines, small protrusions of a dendrite here. This is fluorescence lifetime imaging of a dendrite, and this is a dendritic spine, neighboring dendritic spines, and so on, to be one of the key hallmarks of learning and retrieving memory in animals. So this is the motivations that why we want to study the actin reorganizations in dendritic spine. So let me start with the image that you've seen this on the left here. So at around 0 seconds, the neuroreceptors will release the white arrowhead here at 0 seconds. You see that the glutamate is uncaged. And then once the glutamate is uncaged, once the neurotransmitter is rushed in through this dendritic spine, you see that the volume increases and the shape changes. Now, there are multiple things that I want to say here, but one of the more important thing is that while dendritic spines plasticity, they work together. However, you can see that if I particularly stimulate one dendritic spine, you see that the neighboring dendritic spine doesn't react. So how does a dendritic spine form? Well, it starts with the filopodia initiations, and then you see that it elongates and it forms a small spine head. And then as a mature dendritic spine, you see that there are two compartments here. One is the head, and then the second part is the neck. You see that there are multiple actin binding proteins in these cartoon representations of actomyosin network. For example, the main topic of this talk today is the ARP23 complex, and there's also myosin 2, multiple actin cross-linking protein, cofilin, and so on. Now, ARP23 complex, like I mentioned in the title, is a filament brancher. It's an actin filament brancher. But not only it branches, but also behaves as a nucleation site. You see that when I have a mother filament, ARP23 complex will nucleate daughter filaments, and the angle between daughter filament and its mother filament is approximately 70 degrees. So you see that this ARP23 complex works as a gives the structures to the network, but also we want to learn how it shapes the dynamics of actomyosin networks. So the objectives of this talk today is to understand the role of branching in cytoskeletal networks. And the second one is to understand marginal stability and avalances in cytoskeleton. So the avalances are rare events that we see in our simulations, and I will discuss that much more in details. And the animations on the right is a sample of what kind of simulations that we did. So let me tell you how we model our simulations. We have used and modified a powerful mechanochemical model of actomyosin systems called MEDIAN. MEDIAN is actively developed by Dr. Popoyan and his group at University of Maryland. I think my friend Carl from Popoyan's group will give a talk on Wednesday. So in this example, you see that I'll just go over the components of my network. I have a long red cylinder, which is an actin filament. I have a long moving blue cylinder here is the walking motor, non-myosin two-way motor, and then I have a short cylinder representing a green short cylinder representing alpha-actin and linker. Now, a couple of things I want to mention here is the simulation space that we have is a rigid box, is a rigid boundary box, meaning that the box has this repulsion potential to make sure that everything, for example, actin filament here, is inside the space, the simulation space. And this is particularly important for the next slide and the next couple slides, because that we can calculate the shortest distance between an actin filament to each boundary. And this is an example of a branch Actin-myosin network. The difference here is that we added R23 complex, a filament brancher is represented by yellow bit. As you can see, that there's a yellow bit. Now, if I'll just run it until around 1300 seconds, around 1300 seconds, you will see a large shift displacement in the network, and that's what we call avalanches. And I'll go into more details in the next couple of slides. Also, to mention median here, median represents not only stochastic chemical reactions, but also the deterministic mechanical representations of actin filaments and the components of the Actin-myosin network. However, maybe more importantly, it also has this mechanochemical feedback that allows for us to observe the dynamics of the Actin-myosin network. So let me give you one of the first results that we got. So we were able to observe that contractility requires motors and linkers. Now, since I mentioned that my simulation space is a rigid box boundary, we can calculate the closest distance of every single actin filament to each boundary. Therefore, we can calculate the boundary interaction energy of the total of the system with respect to the boundary. Now, this state diagram here represents that, the boundary interaction energy. The x-axis here is the ratio of linker to actin. This is the same x-axis, and then the y-axis is also ratio of motor to actin. Now, as you can see in this region highlighted by the green dash box, that also the color map here is that the more red, or the redder the color, means that the boundary interaction is higher, and then the whiter the color here means the boundary interaction energy is lower. Now you can see that in these particular regions, we have a network where it is less contracted, more contracted, and then less contracted. We call this a re-entrant phase of the contractility, and it has been shown experimentally. So this is the from 2008 from Dr. Waite's group. And you see that with the same x-axis and y-axis, you see that this is an experiment on constituted actin solutions. And the symbol here, cross, means the system is contracted, and circle means the system doesn't contract. And the gray shaded area here is their interpretations, or where the locations of the contracted system is. And you see that there is this re-entrant phase of the contractility. Now, I need to mention that our group in 2012 has also shown that contractility requires motors and linker by previous model without accounting for actin treadmilling. In our model that I'm showing you here is we have actin polymerization and depolymerization, therefore we can also model actin treadmilling. You see the same behavior that I mentioned, a re-entrant of contractility. Now, let me describe the avalanches that I show you a couple slides back. Now, I need to quantify these avalanches. So avalanches, like I mentioned, is a sudden large shift of actin filaments that we observe in branch Actin-myosin networks, and while we do not observe in unbranched Actin-myosin network within our simulation parameters. So these two figures here are describing the same trajectory of simulations. I have three order parameters. The first one is this ratio of radius of gyrations or compression factor, and I have average filament displacement, and I have this delta S, delta shape. So you see that this compression factor can identify one type of an avalanche, which we call a compression here, and then the other delta shape can identify a shear avalanche. Now, average filament displacement can identify these two, while the delta RG and the delta S can give you types of these avalanches. So here is what avalanches looks like in our simulations. You see in this particular simulations, I have a before avalanche and after an avalanche. You see that there is a shift, huge shift of filament in this area, and then you see the basically the actin filaments are contracted inside here. So you see that it moves inside. So we track the tensions in these snapshots. You see that I have a two high tension hotspots in these snapshots. And here is this nice, this is my last slide, animations of the animations of change in tensions. So, okay, so we are showing from 440 seconds to 450 seconds. You see that the high tensions, there are two high tension hotspots here. And at 450 seconds, which is after an avalanche, this particular tensions hotspot here disappears, which we call as epicenter. So here we show you that while the epicenter is here, that the average or the highest filament displacement is elsewhere. So I hope that I have shown you that the branch networks show the behavior of marginal stability state that reminiscence of JM granular states or colloidal gases. And everything that I mentioned is in this paper that we recently published, and I'm happy to take some questions if you have any.
All right, thanks a lot, James. We don't have any in the Q&A yet.
I have a question, if you don't mind.
Please.
So this is all very good, but how do I know that this has anything to do with actually the biological system?
So that's a really good question. So when we observe this in our simulations, we look up different experimental papers that basically see in a step-like displacement in Actin-myosin network. So let me explain that. That experimentalist, Dr. Freitberg from Harvard Public Health, says that when he put a bead on a cortex actin here on top of the surface of an Actin-myosin network, and he tracks this bead, he sees not a random movement, but a large and step-like displacement. He called this event or phenomenon as cytoquake. So we believe that we explain what is the possible mechanisms of this cytoquake. And I believe Carl will also explain it on, I think, Wednesday, if I'm not mistaken, how he did his Haitian analysis on the cytoquake. So yeah, so in 2008, I think cytoquake has been observed, and I think this explains what happens in that cytoquake.
Thank you.
Thank you.
All right. I think, yeah, we're just about out of time. So
I have a question, quick question.
Please go ahead.
Arpita. Yeah. Do you have an idea of what the frequency of these avalanches are and what kind of parameters that it might depend upon, molecular compositions, etc.?
Yeah, so maybe during our talk later, I can show you, we plot the distributions of avalanches in the, not distribution of avalanches, but distributions of cluster, actin cluster distributions in both unbranched and branched networks. So this depends highly on whether your system has certain concentrations of R23, whether you have sufficient concentrations of R23 or not. Because then when you have a sufficient concentration of R23, your network is basically consists of short actin filaments. This makes the motor then not able to walk as much as when it's in the unbranched network. Does that answer your questions?
Yes, yeah. So I'll join the chat later. Thank you.
Thank you.
All right. So thanks again, James, for the wonderful talk and interesting discussion. So moving on to our next speaker, Rohit Vaidya from UIUC. So Rohit, please go ahead.
Hello, everyone. So I think you all can see my screen.
Yes.
So I'll begin. I'm a biophysics PhD student in Professor Paul Selvin's lab at UIUC. And today I'll be talking about a diagnostics technique that we are working on for COVID-19 in our lab. And so I guess this topic needs no introduction. This is the reason we are all meeting virtually and not in person. So this is a novel coronavirus that first originated in December 2019 and has already spread rapidly almost everywhere in the world, has infected millions of people, causing the disease COVID-19. And it has also resulted in hundreds of thousands of deaths so far. So the basic architecture of this virus is like this. So there are three integral membrane proteins, M protein, E protein, and S protein. The S protein, or the spike protein, is the one that gives its distinctive feature, the spikes or the crown, the corona. And inside it, there is the genetic material, RNA, and a nucleocapsid protein that surrounds it. Currently, the only effective way for us to prevent the spread of the infection is to test and isolate, to test as many people as possible and isolate the infected ones. There are three general approaches to testing for this disease. First involves detecting genetic material. Then another approach that involves detecting the antibodies that the host has produced while fighting against this virus. And also then there is antigen detection, which involves detecting viral proteins. So for detecting genetic material, the current gold standard is RT-PCR. And it's very sensitive, very accurate, but it's not quick. It takes several hours, involves multiple heating and cooling cycles. So it requires like a thermocycler or a centralized facility where we can do it. And also it requires sample pretreatment, which can lose or damage the DNA, resulting in false negatives. For the antibody detection, it is fast, but it's much less sensitive than RT-PCR. And it also provides whether you have the virus or you have had the virus, but it doesn't really tell you with very good accuracy whether what state of infection you currently are at. Finally, we come to the antigen detection technique. Currently, there's just one FDA-approved method. It is fast, but it's less sensitive than PCR, so it results in false negatives. And a negative result with this method needs to be verified with PCR again. And it detects nucleocapsid protein using monoclonal antibodies. So it requires, again, pretreatment for breaking open the virus. And it can result in further false negatives. So we thought, what if we could detect the virus without having to do any pretreatment? If we could just detect the virus based on its membrane proteins. And the candidate that we thought was a good one was spike protein. So this is a cryo-EM structure for the spike protein here. It usually exists as a tetramer, and it plays a crucial part in recognizing and entering the host cell by binding to ACE2 receptor. So you can see a picture here of the human ACE2 receptor binding to the SARS-CoV-2 receptor binding motif. if we can devise an antigen test to detect this receptor binding motif or receptor binding domain of the spike protein, it could tell us directly about the amount of virus. Not only that, we could use that for looking at the infectivity of the virus. So the method for that that we devised, the schematics looks like this. You can see the virus here with its triangular spikes. And we use a DNA aptamer for this. It's basically a single-stranded DNA. And we use something called rolling circle amplification to achieve higher sensitivity. The benefits of using an aptamer is that it can be designed to be very specific to a certain protein, but just like an antibody, but it can, it's much smaller, and it's much easier to synthesize because it's usually like a 50 nucleotide single-stranded DNA. In our method, we use a complementary strand to bind to the aptamer. So when there is no spike protein present in the solution, the aptamer exists in a double-stranded state. But when the virus or the spike protein is present, the aptamer is designed, or the complementary strand is designed in such a way that the aptamer prefers to bind to the spike protein more than the complementary strand. So it detaches and binds to the spike protein. So the dissociated complementary strand is then left by itself. We use a circular template to which this complementary strand can then go and bind. And we add 529 polymerase that then amplifies this complementary strand, which is now acting as a primer. And this is basically the rolling circle amplification. And it can go on and on amplifying it depending on the time and temperature. And we use a Fret hairpin probe to detect this. So we are using a Sci-Fi and black hole quencher as Fret pairs. So the isothermal nature of RCA makes it simpler compared to the PCR, where it has multiple heating and cooling cycles. The amplification that we get is almost 5,000-fold in 2 hours at 30 degrees Celsius, or if you could heat it up to 42 degrees Celsius in 1 hour, we can get up to 6,000-fold amplification. And also, this is just for one spike protein. There are up to 70 spike proteins for one virus, so the amplification is that much more. So the candidate for the aptamer that we chose to test first was based on a preprint from a Chinese group. So these are the two aptamers that they proposed. They did some experiments to show that they bind, and also they did some molecular docking studies to show that it binds to the receptor binding domain of the spike protein. So we decided to test these ourselves. And for this, we use something called fluorescence anisotropy. So fluorescence anisotropy basically involves looking at the parallel and perpendicular polarizations of the fluorophore and then calculating the anisotropy from that. So essentially, when the fluorophore is free to rotate, the fluorescence is ideally completely isotropic, so anisotropy is low. So here you can see that when it's bound to a DNA aptamer and which is free to rotate, the anisotropy is low. But in its bound state, it is locked in a conformation so that the anisotropy increases. And using that, we basically took 1 nanomolar of the aptamer and added different concentrations of the receptor binding domain of the spike protein. And from this, we could get curves like this. So the anisotropy basically increases and then sort of plateaus. And from that, we calculated dissociation coefficients for this for these aptamers. And for the first aptamer, we got about 40 nanomolar. For the second aptamer, we could get about 27 nanomolar. So this was fine. Then since this was a protein in solution, we decided to test it on a membrane anchored protein to see if that makes a difference. It could. So since we don't have the actual virus, to mimic this, we expressed the protein in human cells, and we looked at the surface. So with immunostaining, we can confirm that the spike protein is getting expressed in these cells. So in the red, you can see that we have targeted the receptor binding domain of the spike protein. In the green, you can see that we have targeted the S2 domain of the spike protein, and they co-localize well. And these are nuclear stains in blue. So we can confirm that the S2 region and the receptor binding domain regions of the spike protein are expressed and accessible for binding. But unfortunately, the aptamers don't seem to work. The aptamers that we were testing in these cells for the protein expressed in this way. So we are at this point right now, and we see that the aptamers characterized using the free RBD protein do not work in the membrane-anchored glycoprotein, at least the ones expressed in human cells. We are also working on testing these aptamers on a pseudovirus. Then also parallelly, new aptamer development is underway in Professor Yilu's group here at Illinois. And that will be based on binding with the membrane-anchored protein. And for selection, they're using pseudovirus. And also, we are parallelly developing a technique using amplification, but with anti-RBD antibody, using something similar to a protein ligation assay. And so with that, I'd like to thank the Selvin group. Even though I'm the one presenting today, everyone in our group is working on this project for over a month now. And so like people are doing experiments as we speak. I'd also like to thank Professor Yilu and his postdoc, Dr. Anna, who are currently working on developing new aptamers, and Dr. Xing Wang for his helpful discussions, and also the sponsors, NSF and CPLC. And thank you. I'd like to thank the organizers for allowing me to talk today. Thank you so much, and I'll take any questions.
All right. Thanks, Rohit. Do we have any questions from the panelists?
Yes, it's Herbie Levine. Can I just ask about maybe obviously you don't have the experience in this case, but in general, how much do these aptamer methods suffer from cross-reactivity, that you would just detect other coronaviruses because their spikes are similar? Do we know much about that?
So we have not, so no one has tested these aptamers with other coronaviruses right now, but I know that with antibodies, currently there are almost no antibodies that can differentiate between the original SARS virus and the SARS-CoV-2. In fact, most of the antibodies that are produced, that the companies are commercially producing right now, they were first produced for the SARS virus, and they just tested it for these viruses and they seem to work. So for Aptamer, I'm not exactly sure.
Okay, other questions?
I had a quick one, Rohit. So you would actually like to test this on the real virus, but what sort of lab does that require for you in order to do that?
Actually, yeah, to handle the real virus, we need a biosafety level 3 lab. And those are very, accessing those labs is very difficult, at least in the University of Illinois system. there's just one lab in Chicago.
Don't they have any deactivate virus or something like that? Or
Right, yeah. So we have also ordered from CDC heat inactivated viruses from NIH actually, heat inactivated viruses, and the problem with that is they are very limited in amount.
So yeah, and then just to get back to that, so the problem that with the virus testing that everybody's doing right now is, I think you said you have to take the virus like from from your saliva. I think you put it in a soap solution or something to break open the cell to get at the RNA.
Right, yeah.
Is that where it gets contaminated? What's wrong with those tests exactly?
So here, people have observed that during the pre-treatment, there is a chance of either losing or damaging the RNA. And in some studies, people have also observed contaminations when when people were working with mass testing, working with mass testing facilities. So like the more steps you involve, the more there is chance for errors.
Okay.
All right. So we are now a little bit over time. So thanks again, Rohit, for the very fascinating and timely talk.
Thank you.
So our last speaker of the day is Asim Hassan from Northeastern University.
Yes. Hi, everyone. So,
hey.
Can you all see my screen now?
Yes.
Mm-hmm.
Okay, very good, very good. Okay, okay. Hi, everyone. I will start by thanking the organizers for giving me this opportunity to share my research with you today. So my name is Asim Hassan. I'm a graduate student working in Northeastern University with Paul Whitford. We are studying the ribosome. And today I will be discussing using simulations to identify precise single molecule probes for ribosome dynamics. So the ribosome is a complex ribonucleoprotein machine that is present in all.
Sorry to interrupt. Awesome. I assume it's everybody, but so we're seeing your slide view. Can you do to do
presentation mode?
Yeah. Thank you. Present.
Oh, okay. So is this better now?
No, you have to change which screen you're sharing.
Oh, really? Okay. Is this good now?
I don't see anything now, actually.
Oh, really? Okay. I'm not sure what's happening here. Yeah, this is the way I'm just sharing my desktop.
Have you gone to presentation mode in PowerPoint?
Yes, this is not PowerPoint, so this is just no, this is not it.
Well, I guess you can go back to what you were doing initially, because then
Yeah.
and then just move it by hand to the next screen.
Yeah.
Okay. So you can see the PowerPoint now, right? But you cannot see the full screen, you're saying?
Right.
Okay, so I will try to do this one last time. If not, I will just go through the slides.
Hey, that worked.
Okay.
I think so. Well, it kicked out again.
Yeah.
Okay. Okay, so this looks good right now. So
okay, So, okay, so sorry for this technical difficulties. Okay. So as I said, my name is Asim Hassan. I'm working in Northeastern University with Paul Whitford on the ribosome, and I will be discussing using simulations to identify precise single molecule probes for ribosome dynamics. So you can see the next slide now, right?
Yes. Yeah.
Okay, perfect. So the ribosome is a complex ribonucleoprotein machine that is present in all prokaryotic and eukaryotic cells. It is responsible for synthesizing all the proteins inside all cells. So it is formed of two subunits, the large subunit and the small subunit. Each one of these subunits has three different binding sites for tRNA molecules. So these are the A site, the P site, and the E site for each subunit. These tRNA molecules are the molecules that bring the amino acids to the newly growing peptide, the newly growing polypeptide chain that will fold into the new protein after the translation process ends. So and each newly coming amino acid on a tRNA molecule is being incorporated in an elongation cycle in this ribosome. So of course, the ribosome is a very huge molecular system. It's very computationally very expensive to study, and we face the usual difficulties when trying to describe large systems like these. So the dynamics are very complicated with lots of coupled motions. There are a lot of experimental difficulties in preparing these systems or studying their dynamics using SMFRET experiments, for example. It's also very difficult usually to analyze the experimental data and to separate them from the noise. It's not easy at all to choose reaction coordinates or to construct free energy landscapes for the different dynamical events in the ribosome. And on top of that, characterizing the barriers, whether they are entropic barriers or energetic barriers, characterizing the transition state ensemble is very difficult. So we have used the structure-based models to study the dynamics of the ribosome. So structure-based models, or SBMs, have originally been developed in the protein folding field and to study protein dynamics also, but they can be used to study native basin dynamics of any biomolecular assembly. And the common feature of all SBMs is that we use the experimentally verified structures, either from X-ray crystallography or cryo-EM models or any other experimentally verified structure, to define some or all of the stabilizing interactions in the force field. So these are the usual SMOG force fields, or the structure SBM force fields. We have the usual covalent bond geometry terms, the bonds, the angles, the improper dihedral angles, and the dihedral term. We have also the Lennard-Jones-like interaction potential between all of the atoms that are in close proximity to each other in the experimentally verified structure, and these define the contacts for the force field. And on top of that, we have the steric repulsion a term between all of the atoms that are not in close proximity to each other. So this represents the exclude volume interactions. So this Lennard-Jones-like interaction will just represent all of the stabilizing interactions in the native basin. And we will be investigating the dynamics around this native basin or in between different basins in dual basin or multi-basin models. And it's also worth mentioning that we have represented all of the atoms in this study. So this is an all atom simulation. It's not a coarse-grained simulation. So we have used the structure-based models to study the translocation step in the elongation cycle. So the translocation step is when the A site tRNA and the P site tRNA translocate to the P site and the E site respectively. So during this, and this is to evacuate the A site for a newly coming tRNA to go into another cycle of elongation. So during this translocation of tRNAs, major structural changes occur in the ribosome. So the whole small subunit rotates with respect to the large subunit, and on top of that, the head domain of the small subunit rotates with respect to the body, and it also exhibits a tilting motion towards and away from the subunit interface, where during which the translocation occurs, the tRNA translocation occurs. So we have used Euler angles to define the head rotation angle and the head tilting angle during translocation from a reference plane, and using these Euler angles as rotational coordinates allowed us to follow the structural changes in the head domain during translocation. And we have also employed a dual Bayesian structure-based model to study the translocation step between the pre-translocation state, where the both of the tRNAs are in the A site and the P site, and the post-translational state, where post-translocation state, where the two tRNAs are in the P and the E site. So we have used these two experimentally verified structures to define the potential energy minima for the for our force field, and we didn't say anything about any other intermediates. So we were kind of investigating the energy landscape between these two minima during translocation. So in order to monitor the translocation events, we have used the tRNA reaction coordinates to monitor the translocation. So we started all of the simulations from a partially rotated hybrid state, the A/P, P/E state. And then translocation events occur spontaneously, where the tRNA coordinates reach the value of 0, indicating the end of translocation. And during this representative translocation event, the head starts by rotating and then back rotating. Also, the head tilts and back tilts. So these structural changes in the head domain, as we can see, are highly coupled together, and it's very difficult to disentangle between these two motions dynamically, or to detect them separately in asymptotic experiments, for example. And I will address that shortly. So if we investigate the 2D probability distributions of the system during translocation in this 2D configuration space. So here, this is showing the 2D configuration space of one tRNA reaction coordinate versus the head rotation angle of the head of the small subunit. This shows very distinct peaks. So this one is the hybrid state peak. This is the post state where translocation has ended, and we can see very distinct intermediate peaks. And it's very evident that although the system is in the force field, we didn't specify anything about the intermediate states, but more or less the ribosome is a crowded system, so that the steric interactions that have been built in the force field dictate this narrow pathway in this 2D configuration space that the ribosome has to pass through it in order to finish the translocation step. And if we consider the 2D configuration space with respect to the tilting angle of the head domain, we find that in order for the translocation to end in the post state, the head has to tilt to around 10 degrees or so. And all of these peaks are actually, they correspond to experimentally verified structures in cryo-EM models. On top of that, we can use the structure-based models to get an idea about the factors that lead to the head tilting. So examining the simulations, we found that the steric repulsion or steric interactions between the P site tRNA and the PE loop and S13 protein of the small subunit, these were probably the reason for head tilting. And actually, when we removed the steric interactions between these two regions and the P site tRNA, this has decreased the range of head tilting while not affecting the range of head rotation during translocation. So the translocation event occurs, but the head doesn't tilt that much. So now emerges a question: is that why is it difficult for studies of dynamics like in asymptotic experiments, for example? Why is it difficult to detect head tilting while at the same time it's always reporting head rotation? So, for example, here in this study that was done in the Blanchard laboratory, where they labeled the two proteins, one in the large subunit, another one in the small subunit. And in another instance, they labeled the P site tRNA and the S13 protein of the small subunit. They have detected the FRET signals changes during translocation, and they have interpreted these changes in terms of head rotation or exaggerated head rotation. But they also stated that head tilting or other forms of head rotatory motion cannot be ruled out by these studies, and that the coupling between head rotation and other modes of rotation, like tilting, for example, they need quantitative molecular dynamical simulations in order to separate them. So in order for us to address this of how to separate between head rotation and head tilting, we have examined all of the pairwise distances between all of the proteins in the small subunit of the ribosome. So these are 20 proteins in number, 10 in the head domain, and 8 proteins in the head domain. and 12 proteins in the body. So of course, since these are nearly rigid bodies, all of the intra-domain distances will not be correlated with either head rotation or head tilting, but we doubted that or suspected that some of the distances between a protein in the head domain, for example, and another protein in the body, they could be correlated with either head rotation or head tilting. So this is showing the mean correlation of a specific distance with head rotation versus the mean correlation of that specific distance with head tilting. And as expected, most of the distances are either highly correlated or anti-correlated with both of head rotation and head tilting, because these two motions are highly coupled. But more interestingly, there is a group of distances in the small subunit that shows a high degree of correlation with head rotation, but not that much with head tilting, or vice versa. And when we examined these distances, we found that all of the distances that are specific to head rotation, and they show a high degree of correlation with head rotation, but not that much with head tilting, all of these distances are parallel to the axis of head tilting. While on the other hand, all of the distances that are specific to head tilting and they don't correlate much with head rotation, they are parallel to the axis of head rotation. And this follows the intuition, you know. So investigating the 2D histograms has revealed a more complicated story. So of course, these are rotatory motions, and it will show definitely nonlinear dependencies between the distances and the structural coordinate, either rotation, head rotation, or head tilting angles. So here, for example, this is showing a 2D histogram of a certain distance and either head rotation or head tilting, and we can see the nonlinear signals very clearly. So we were able to divide all of the distances in the small subunit into 5 different groups, depending on the qualitative nature of these 2D histograms. And in order for us to address these nonlinearities, we have used the mutual information as a way to probe these dependencies that are nonlinear. So the mutual information is defined to be the KL divergence between the joint probability distribution of 2 random variables and the product of the marginal probability distributions. It measures the mutual information, measures the decrease of uncertainty in knowing one random variable, how much uncertainty has decreased in knowing that random variable. So when the mutual information is 0, this shows that the 2 random variables are probably independent, and when there is any sort of relation between them, are either linear or nonlinear, this will be captured by a positive value of a mutual information.
So awesome. Sorry, you're running a little bit over into your question time. Can you start to wrap up?
Yes, I just have two slides to go.
Okay.
Yes. So here, this shows all of the mutual information of all of the distances with either head rotation or head tilting. And as suspected, these nonlinearities didn't affect the result that much. So there are some distances that can be used to measure head rotation specifically, and not and differentiate between it and tilting. And currently, we are investigating adding other or considering other pairs of distances, not only a single distance. And we are suspecting that knowing another distance at the same time will move some of these points towards the x-axis, for example, where they can be used to detect head rotation more specifically, and not head tilting, or vice versa, move these set of points to the y-axis. So in conclusion, I conclude by saying that structure-based models are very useful to explore native Bayesian dynamics in large biomolecular assemblies. Their simplicity is what grants the simplistic energetic description of them is what grants them explanatory power in dissecting the various energetic and entropic contributions to the energy barriers in transitions, and that integrating experimental and theoretical insights will help in bridging detailed biochemical interactions with large-scale dynamics, and that this line of work can help in designing future asymptotic experiments on the ribosome to differentiate between head rotation and head tilting and other highly coupled motions of the small subunit domains. So finally, I thank my advisor, Paul Whitford, for all of his support. Thanks to Mariana and Prasad also for all of their useful discussions related to this project. Northeastern University Computational Resources, NSF for financial support, and thank you all for your attention and your patience, and I would be happy to answer any questions.
All right, thanks, Awesome. Do we have maybe a question from the faculty?
I have a fast question for you.
Yes.
The regular probes measuring this tilt mode, where are they on that spectrum, basically? You had a very big spectrum where you say what a better distance to measure. The current ones are where?
So all of the experimental probes that have been tried in asymptotic experiments, they didn't report tilting at all. So the nearest study that that reported tilting was this study, and all of the probes that they have used, which are these two, the S13L5 protein and S13-ptRNA, they weren't as successful in detecting head tilting. And when we examined these distances, for example, in our plots, we found that, so for example, here in the mutual information plot, we found that these distances that were used in the experimental structure, they weren't as near to these to this green point, for example, that this green point corresponds to S15, S19. So we believe that these distances are much better suited given the geometry and the dynamics of the ribosome to detect head tilting specifically. But their distances were way further, so they showed a much less mutual information with head tilting.
Okay, thank you.
You're welcome. Thank you.
Could I, this is David Thurmula. Could I ask a more simple question? I think your predictions are really very interesting. I was wondering if you could suggest an experiment where you cross-link the head tilting and computationally examine the consequences, and then ask the experimentalists to do a similar experiment, because putting the probes, as Jose was pointing out, could have some issues, but cross-link through cysteine are much easier to do. In any event, you can answer that question very precisely using your computations.
Yes, I totally agree. So that the whole motivation for this study was the initial paper earlier this year from Robin Gonzalez, where they reported that they can easily use site-directed mutagenesis to insert dyes in any protein. So they kind of liberated themselves from the need of having cysteine residues in these proteins to be able to label them. So based on that, we just proposed that labeling a certain specific set of proteins would be helpful in differentiating the tilting from rotation, for example.
Thanks.
All right. I think with that, we need to wrap up. So thanks again, Asam, for closing the session. And thanks everybody for attending today for the first day. So any remaining questions, just a reminder, you can ask them in the spatial chat. I think the link was in one of your emails. And otherwise, I guess we will see everybody again virtually tomorrow.
JC, can you hear me?
Yes, Dan.
Dan Goldman here. I just wanted to thank JC for stepping into the moderator role today, and thank everybody for virtually attending, and hopefully you'll be able to visit us at Georgia Tech in Atlanta in physical presence before long, and also to thank Margie, Aaron, Mari here at Georgia Tech, among others, Margie at Rice, for all the incredible effort it took to get this meeting going. So thank you all, and see you tomorrow.
Here's Jose Onushik again. Just remind the people that on Tuesday and on Thursday, we're going to have the poster session. Thanks for the people presenting posters. And if you have a poster, I think you still can contact Margie and put a poster now. So all the students that want to participate, please be part of it.
So where is the so-called question and answer session?
It's on the email. There is the link.
A link? Okay.
On the general email for the conference.
All right. Thanks.
Okay.
Thank you, everyone.
Bye then.